Welcome everyone, and thank you for joining us for our presentation titled Detection and Monitoring of Mutations from Liquid Biopsies, a Clinical Study. It's my pleasure to introduce you to Monika Seidel, the Senior Development Scientist from the Genomics and Cellular Research at Sativa. For a complete biography of our speaker, please visit the tab at the top of your screen. Now, if any questions arise during the presentation, we encourage you to submit them through the Q&A box. Our speaker will address your questions following the end of the presentation. Please join me now in welcoming Monika Seidel. I will now turn the presentation over to her. Welcome, Monika.
Good morning and good afternoon, everyone. Thank you for the introduction. My name is Monika Seidel, and I'm part of R&D team at Sativa in Genomic and Diagnostic Solution business segment. Our mission at Sativa is to accelerate and advance therapeutics, and by doing so, we believe that we are getting closer to the situation where access to life-changing therapies is widely available. How do we achieve our mission? We achieve our mission by providing the customers with tools and services for diagnostic-related research, and we also maintain a wide network of collaboration, including academic institutions where this kind of research is taking place. Today, I will be talking about liquid biopsies and their application in cancer diagnostic. This is a rapidly growing field fueled by current focus on development and advances in precision medicine. So what would be covered in my presentation? As an introduction, I will briefly cover liquid biopsies, and then I will describe different types of nucleic acids that can be found in the bloodstream, and they have obviously different relevance and applicability into biomarker profiling. Then we will dive a bit deeper into a cell-free DNA nucleic acid, and this is the only type of analyte that is currently used in clinical practice. We will consider challenges associated with liquid biopsies, and we also will discuss some avenues that are being undertaken in order to overcome those challenges. The core of my presentation will focus on results of a study that we conducted in which we tried to answer the question whether site selection assisted extraction of cell-free DNA can allow for higher sensitivity of cancer-associated mutation detection. The term liquid biopsy covers the collection of bodily fluids for the purpose of genetic profiling. And there's a number of different bodily fluids that can be collected for this purpose, including urine, cerebrospinal fluid, saliva. But this is the blood that is being currently used in clinical practice and is mostly investigated. The first report describing the presence of nucleic acids in the bloodstream goes back to 1948. However, at this particular point in time, the discovery was completely unnoticed and put to rest for roughly another 15 years, when another report showed that fetal DNA can be found and detected in maternal plasma. This discovery laid the foundation for non-invasive prenatal screening that is currently being used in clinical practice for detection of chromosomal aneuploidy in pregnancies. And this amazing success that the liquid biopsies achieve in this field triggered extensive research into the possibility of using liquid biopsies for biomarker profiling in cancer, cardiovascular disorders, autoimmune diseases, diabetes, or for early detection of organ rejection in transplantology. Excuse me. As for cancer related use for liquid biopsies, their applicability is being researched in both early and advanced stage cancer. And for early stage cancer, it's mostly evaluated in terms of being a screening tool. So we are trying to find out whether liquid biopsies are able to detect cancer in otherwise healthy individuals. So in those patients that do not present any clinical manifestation of the disease, but cancer is already developing. Although there is one FDA-approved test for screening of early cancer, this is a very challenging field, and this is mostly due to the limited sensitivity of detecting cancer-associated mutations. Because in an early stage cancer, the amount of tumor-derived fraction in the circulation is extremely low. The reported sensitivity varies depending on the cancer type and cancer location between 30 and 80%, meaning that the risk of false negative results is extremely high. Similar challenges apply to the use of liquid biopsies in the detection of minimal residual disease. So this would correspond to the detection of cancers. The much higher success rate for liquid biopsies is found in advanced cancer, and this is reflected in the current guidelines advocating the use of liquid biopsies for genetic profiling in circumstances where tissue biopsy is not available. And this applies to advanced cancers such as lung cancer, prostate cancer, or breast cancer. Another field which holds a lot of promise for liquid biopsies is for monitoring purposes. And this will be monitoring how well the therapy is working, and also monitoring and detection patients, and unfortunately happen in the majority of patients following the first line of therapy. What needs to be said is the biggest advantage of liquid biopsy is its potential to uncover the whole genetic landscape of the tumor. All happen in the tumor in a spatial and temporal manner. And for that reason, there's a number of different cell-free nucleic acids that can be found in the circulation. If we concentrate on RNA, there are two main groups. One is non-coding RNA, and the second is coding RNA. So coding RNA is represented by messenger RNA, which encodes protein. This is a minor fraction in the circulation, and in its intact form is almost exclusively found in exosomes. The reason behind it is when mRNA is released into the bloodstream, it's rapidly degraded by nucleases, and for that reason constitutes a rather unsuitable analyte for biomarker profiling. Among non-coding RNAs, which is a very diverse set of molecules, but majority of them, with a few exceptions, are responsible for gene expression modulation, it is the microRNA which attracted the highest amount of attention. Those are small molecules of around 18 to 24 base pairs long. They are quite stable in the circulation due to the fact that they are being in complexes with protein. They are responsible for translational silencing of genes, and this is done through microRNA binding to 3' untranslated region in their messenger RNAs. So one microRNA can target a number of messenger RNAs, and this is because this interaction happens for non-full complementarity of two strands. And this is a heavily researched area. Number of reports show a specific cancer-related signature profile of microRNA expression. Now, if we go into DNA, we can divide the DNA found into the circulation based on its origin. So it's either mitochondrial DNA or cell-free DNA that is represented by genomic DNA released by a number of cells in the organism. Mitochondrial DNA research is in infancy, and it's not currently used in any of the analytical methods for biomarker profiling, although there are a number of reports showing that it's exclusively released by tumor cells and might be used for cancer detection. On the contrary, cell-free DNA is already in clinical practice, is used for non-invasive prenatal screening, is used in circumstances where tissue biopsy is not feasible in advanced cancer, and it's also used in cancer screening for colorectal cancer in patients which are unable to undergo colonoscopy. Diving deeper into cell-free DNA characteristics, first of all, it is released into the bloodstream through active and passive mechanisms, and those active mechanisms are not fully understood. As for passive release, is mostly done by the way of necrosis and apoptosis. Cell-free DNA represents the pool of genomic DNA released by a number of different cells, including inflamed cells, tumor cells, fetal cells in pregnancy. But it is believed that the majority of cell-free DNA in the bloodstream is of hematopoietic origin. In terms of the size of cell-free DNA, the peak is centered around 170 base pairs. This is corresponding to the size of the nucleosome. And this size profile of the cell-free DNA, you can see on the right-hand side of the slide. This is an electrophoregram created with the use of bioanalyzer, and you show that the size of cell-free DNA ranges between 100 to 300 base pairs, but indeed the peak is around 170 base pairs. There is a number of reports showing that cell-free DNA is fragmented in a different way depending on its origin. And what is also important, and initially this was something that was considered to use in cancer screening and detection, is that the amount of cell-free DNA is highly increased in advanced cancer. However, it turned out that this high amount of cell-free DNA is also associated with other conditions such as pregnancy, autoimmune disorders, or following strenuous exercise. So if you have a look at this slide, which is a very busy slide, and I have no intention to go through all the publications that are being listed in the table, I just wanted to attract the attention of the audience to the fact that cell-free DNA, which is derived from tumor, is highly fragmented. And all those publications that I list in the table give three conclusions. First of all, the fact that the amount of cell-free DNA in advanced cancer is much higher than in healthy individuals. Secondly, that there is a clear shift in the size of cell-free DNA in cancer patients toward lower fragments, somewhere around or somewhere below 150 base pair. And thirdly, that those fragments that are smaller, they are enriched in tumor fraction. So the biggest challenge of cell-free DNA and its utility for mutation detection is sensitivity. This sensitivity, limited sensitivity, originates, of course, from the endpoint detection assay that they're being currently used for biomarker profiling, but it's also inherent to the specific characteristic that cell-free DNA possesses. So first of all, the amount of cell-free DNA in bloodstream, it's particularly low. But what is even lower is the fraction that derives from the tumor. In early stage cancer, this fraction is within 0.01% and goes up to 30% in metastatic advanced cancer. On the top of this, if we want to use liquid biopsies to monitor tumor evolution, you can imagine that those clonal cells within the tumor then constitute an even lower fraction of the tumor itself. What doesn't help the situation is quite often that during plasma processing, there is a significant release of genomic DNA from white blood cells that further dilutes tumor-derived fraction. And what I mentioned earlier is also the fact that cell-free DNA that comes from cancer cells is highly fragmented and also is damaged, meaning that sometimes it's present in a single-stranded form or in double-stranded form where one of the strands is nicked. This imposes a lot of challenges because the majority of currently used next-generation library preparation methods use double-stranded library preps, which unfortunately are not allowing to capture those small fragments and those nicked fragments in the endpoint library and NGS analysis. If we now go to the different technologies that are being used for detection of mutations, I listed two different ones. One is digital droplet PCR, and the second one is targeted NGS. You can see that both of those methods, they have a different limit of detection. And this limit of detection varies depending on some technological changes that are being implemented to the standard technique in order to lower limit of detection. For NGS, for example, that would be use of unique molecular identifiers that would allow to eliminate PCR-created errors, or the use of bioinformatic tools that allow to eliminate sequencing errors. Nevertheless, the first question that everyone will probably ask, if the limit of detection for digital drug with PCR is so low, why isn't it this technique exclusively used for biomarker profiling? And the reason behind this is that although it has extremely low sensitivity, this method requires that the user knows the mutation a priori to the detection. So you need to know what we are looking for when you are using digital PCR. In terms of targeted NGS, this a priori knowledge of mutation identity is not needed. What is needed is the identity of the gene that we want to investigate. And there are a number of different panels that allow sequencing genes that are well known to be associated with cancer. So on the previous slide, I discussed the challenges associated with typical characteristics that cell-free DNA has. And there are a number of avenues that can be undertaken to mitigate those problems. So for example, for the release of genomic DNA and the increased yield in cell-free DNA extraction, it has been now advised to use specific type of blood collection tubes that allow to limit genomic DNA release from white blood cells and increase the stability of cell-free DNA. Again, for library preparation, it has been suggested that in some cases, single-stranded library preparations might be more suitable in order to capture those nicked and single-stranded fragments of DNA. Again, I already mentioned the use of bioinformatic tools to eliminate sequencing error in NGS, and also use of unique molecular identifiers to eliminate PCR amplification errors. There is also another point at which we can try to mitigate the problems related to cell-free DNA, and this is the extraction. So here at Cytiva, we develop cell-extracted cell-free DNA kit that is based on magnetic bead technology and has 3 features that we believe allow to increase the sensitivity in endpoint assay. And those 3 critical points is the fact that the kit allows to capture low fragments in a very efficient way. The kit allows to extract cell-free DNA to the high extent. And the third aspect of the kit is the fact that it allows for elimination of genomic DNA carryover. This principle is illustrated on the electropherogram that you can see on the right-hand side of the slide. So if we have a look, the blue trace represents plasma extracted with our kit, but before extraction, the plasma was spiked with DNA ladder, and this DNA ladder is represented in the green trace. So when you have a look at the efficiency of extraction of small fragments, you can see that the blue line overlaps very well with the green line in lower molecular weight range. For example, for 50 and 100 base pairs. When you have a look at the higher molecular weight recovery, you can see that above 300 base pairs, the recovery declines rapidly. And at 2 kb, this recovery is around 20% or below 20%, meaning that the carryover of genomic DNA is minimized. So we, of course, wanted to see how our kit performs against leading brands. So we benchmark the kit against MacMac cell-free DNA kit, which is manufactured by Thermo Fisher Scientific. And we also tested the kit against QIAMP mini-lot kit, which is manufactured by QIAGEN. So in terms of QIAGEN kit, this is a silica-based kit that is not very easy to be automated. It can only be automated on instruments manufactured by QIAGEN. In terms of MacMac kits, this is a very similar format to Cytiva kit. It's based on magnetic bead technology, which allows for automation. So if we concentrate on the cell-free DNA yield that can be seen on this graph, you can see that there is not much difference in the extract and efficiency between 3 different kits. They're pretty much similar. However, when we move to the genomic DNA carryover, we have an obvious advantage in the field. It is Cytiva kit that allows to significantly reduce the presence of a genomic DNA in the extract obtained from patient serum. Of course, we are very encouraged from those results, but the main question that we ask is whether those key performance characteristics that I just described for our kit allow to achieve higher sensitivity of mutation detection in clinically relevant scenario. And for that reason, so to answer the question, we designed a small study that involved 4 patients that were all diagnosed with an advanced stage lung cancer. They donated blood. This blood was processed and plasma was extracted. The plasma was subjected to library preparation, and tumor profiling was done using targeted NGS. So a bit more about patient stratification and why we decided to go with patients with non-small cell lung cancer. So first of all, this is the cancer that unfortunately is usually detected at a very advanced stage. And the reason behind this, because first of all, there are no screening tests for this cancer. And secondly, the cancer usually is asymptomatic and starts giving symptoms when metastasis already occurred. The genetic profiling is currently used in clinical practice in order to discover mutations that can be targeted with specific type of therapy. And this genetic profiling is a device for testing the mutation and the mutations in 5 genes which are listed in this table. And this includes EGFR, ALK, ROS1, BRAF, and NTRK. All those genes, they code for protein kinases, and all of those protein kinases, they have specific inhibitors can be used to treat patients. What is also important, and this is the other reason why we decided to go for those cohort of patients, is the fact that in advanced lung cancer, in circumstances where tissue biopsy is not available, liquid biopsy is advocated by current clinical guidelines. The next and very important aspect of this cancer is unfortunate resistance that occurs in the majority of patients following first-line target therapy. And this is specifically true from patients that are positive for mutation in EGFR. Usually within 12 months, all of those patients develop secondary resistance. And this secondary resistance is either related to additional mutation in the EGFR gene itself, or through some bypass mechanism that, for example, involves amplification in MET gene. Okay. So the plasma that was obtained from those patients was extracted using 3 kits, and we used the same kits as I discussed on previous slides. So this was the Magmax kit from Thermo Fisher Scientific and QIAGEN kit QAMP, which is based on silica columns. All patients but 1 were processed with all 3 kits. Only patient 1 was processed with Cytiva and QIAGEN kit, and this was due to the limitation in sample volume. The extract were analyzed using capillary electrophoresis on bioanalyzer. And maybe if we first concentrate on the right-hand side of the slide, we see 3 electropherograms. The green trace represents Cytiva extract, the blue trace represents Magmax extract, and the orange one is from QIAGEN. As you can see here, the amount of cell-free DNA, so the main peak centered at around 170 base pair, is similar between 3 different kits. However, if you have a look at genomic DNA peak that would represent anything above 700 base pair, you can see that this is present in both QIAGEN and MAGMAX extracts. However, as far as Cytiva kits, is almost completely absent. When we go to the left-hand side of the slide, this is the summary of the results. So the top graph represents the cell-free DNA yield separated by the patient and by the kit. And even though there's a lot of diversity in the extraction profile, you can see that pretty much overall the performance of all 3 kits is similar. The clear difference is when we go to genomic DNA contamination. This is summarized in the lower graph. You can see that this is the Cytiva kit that allows to limit genomic DNA presence in the extract. Okay. So all of those extracts were subjected to library preparation. And this library preparation that we performed used the total volume of all samples. The targeted sequencing was done using ion torrent technology, and the targeted panel that we use is the panel that was developed by Biocept, and it's called Biocept Target Selector NGS Lung Panel. It allows to sequence 12 genes that are well known to be related to lung cancer. What it needs to be said at this point is that library preparation, sequencing, and data analysis was performed at Biocept. So what you see on the slide, those are results from targeted NGS, and I'm going to discuss those results in detail on subsequent slides, but maybe we can just concentrate and compare the mutation allele frequency between patients and between kits that were used for extraction. As you can see, in all cases, from patient 4 to patient 1, it is a Cytiva kit that allows to detect mutation at the higher frequency. And this obviously indicates that when plasma is processed with our kit, the extract is enriched in tumor-derived fraction. What you can see in the lower table, this is the analytical sensitivity of the target selector, and you can see that for hotspot single nucleotide variants, deletions and insertions, the limit of detection goes to 0.1%. For the novel mutations, this limit of detection is at 0.5%. Okay, let's now try to dive a bit deeper into each of the patients. So let's concentrate on patient 2 and 4. Both of those patients were found positive for mutation in BRAF kinase, and this is the mutation at the position 600, which results in the replacement of valine with aspartic acid. This mutation is a phosphamimetic mutation, means that even though the protein is not phosphorylated, the presence of negatively charged aspartic acids make the protein active. And through this activity, the kinase activates downstream effectors, so it activates MAP kinase, and this signaling leads to the uncontrolled growth and proliferation of the cells. This is a very well-known mutation for melanoma, not that prevalent in non-small cell lung cancer. I think it's only present in 2% of the patients, so quite rare. Nevertheless, which is quite beneficial for the patients, there is targeted therapy. And this targeted therapy is both approved in the European Union and in the United States, and involves the use of 2 drugs targeting 2 different kinases. So the first drug targets BRAF kinase, the second drug targets MAP kinase. And what you can see on the table on the right, this is the table taken from Novartis website, who is a manufacturer of both drugs. You can see the clear benefit for those patients, where you can see that overall survival is increased to 20 to 24 months. So what I wanted to concentrate on those slides, I wanted to analyze again and go slightly deeper into the genomic DNA carryover, the yield of cell-free DNA, and how those two things translate into mutation allele frequency detected in our assay. So when we start from patient 4, you can see that Cytiva is the best in terms of cell-free DNA yield, and of course the best in terms of minimizing genomic DNA contamination. And in this particular instance, the mutation allele frequency detected in Cytiva extract is twice as high as in competitors. But I think what we need to concentrate is patient 2. Have a look at patient 2. Patient 2, the yield extracted is similar between 3 kids. You can see that the amount of cell-free DNA is very low in this patient. And again, you can see that genomic DNA contamination is the lowest in samples extracted with Cytiva kit. However, or maybe what's important is that the mutation allele frequency for extract process with Cytiva is almost 7 times higher than for the competitor's kit. And this brings us only one conclusion, that the reduction in genomic DNA contamination is specifically important in circumstances where cell-free DNA is a limiting factor. Okay, let's now discuss the results for patient 1 and patient 3. They are both found positive for mutation in tumor suppressor protein 53. This is the protein which is quite often mutated in many different cancers. Not so prevalent in non-small cell lung cancer, very prevalent in lung cancer associated with smoking. The protein is composed of 3 domains, with DNA binding domain most important for suppressor activity of this protein, and also this domain is targeted by a number of mutations that are well known associated with cancer. So this is a mutation hotspot. Those mutations occur in conserved residues that are listed on the slide. And one of those conserved residues is mutated in patient 1. Please let me remind you that patient 1 was only processed with 2 kits, QIAGEN and Cytiva, due to the sample limitation. I want to concentrate on patient 3 because this is slightly more controversial and slightly more difficult to explain the results. So when we consider MACMAX kit and the extract from MACMAX kit, we didn't detect any mutation in this extract. When we have a look at the Cytiva kit, it detects the mutation that is one of the hotspot mutation in well-known conserved residue. However, now going to QIAGEN kit, we can see that the text mutation, this mutation is not a hotspot mutation, and the mutation allele frequency is 0.35%. This means that the mutation frequency is unfortunately below the level of detection for novel single nucleotide variants. And in this circumstance, the clinical scenario, this mutation would be considered inconclusive result and would have to be repeated. But please let me remind that there would be no way of repeating it because the whole extract has been already used for targeted sequencing. And now, if we dive slightly deeper in mutation allele frequency, and we will have a look at this frequency considering the cell-free DNA yield and genomic DNA contamination in the sample. Let's concentrate on sample 3, where we don't see any concordance between 3 kits. And as you can see, QIAGEN appears to be the best here in terms of the recovery of cell-free DNA. While Cytiva is quite comparable to MACMAX. The major difference that we see is in genomic DNA contamination. Again, you can see that minimal contamination with Cytiva kit, however, quite high with MACMAX. And let me remind you that this is actually the MACMAX kit in which extract no mutation is being detected. So from those results, we can draw one important conclusion, that size selection-based extraction allows to detect mutation associated with cancer in circumstances where those mutations can be missed when other non-site selection types of extraction are implemented. Okay, so just to summarize the whole presentation, we know that targeted analysis of cell-free DNA is already in clinical practice, is widely used for non-invasive prenatal screening. We also know that it's currently recommended to use cell-free DNA for genetic profiling in advanced cancer in circumstances where tissue biopsy is not available. Very important aspect of liquid biopsies, and the one that holds the highest amount of promise, is the fact that it has the potential to uncover a full genetic landscape of the evolving tumor. So it can allow for comprehensive decision regarding therapy at the beginning of the treatment, but also allows to quickly change the therapeutic approach once the resistance mutation appear. The biggest challenge in the analysis of cell-free DNA is the fact that it constitutes a minor fraction of the circulation, and the fact that cancer-associated mutations are found at an extremely low level. From those experiments that I share with you, it is obvious that site selection-assisted extraction of cell-free DNA allows to enrich for tumor-derived fraction. And this enrichment in tumor-derived fraction allows to detect mutations in circumstances when other kits fail to detect it. Okay, I think this is it for me, and now it's time for questions and answers. Thank you.
Thank you, Monika, for that wonderful presentation. We will now move to the Q&A portion of this presentation. And as a reminder to our audience members, please submit your questions via the Q&A box. Now, Monika, we already have some great questions coming in from our audience members.
Right.
Question is Sera Extracta cell-free DNA kit compatible with serum samples?
Yes, it is compatible. In fact, it's highly recommended for those type of samples. And the reason behind it is that serum samples are notorious for genomic DNA contamination. And as I presented, our kit is able to remove to some extent this genomic DNA contamination.
Thank you so much. Now, in clinical applications, what is a preferable sample type? Is it serum or is it plasma?
I believe that there's a general consensus in the field that plasma is preferred sample in clinical applications, and this is based on a number of reports showing that mutation allele frequency and the detection rate of cancer-associated biomarkers is much higher in plasma samples compared to matched serum samples. What is probably important that we shouldn't dismiss serum samples completely. They are very important in retrospective studies for biomarker discovery in large cohorts of patients on biobank samples.
Thank you so much. And again, I want to thank our audience for these great questions. Now, Monika, is blood always the best type of input sample for cancer detection?
Well, as I said, the amount of dependent on the cancer type, the stage of cancer, the location of the tumor. So there are particular type of cancers that probably are not very well suited to be detected in blood, and this will be bladder and prostate cancer. It appears that urine is more suitable sample. For example, for brain cancers, it seems that cerebrospinal fluid might be more suitable.
Thank you so much. Will liquid biopsy fully replace the tissue biopsy in the near future? Interesting question.
Yes, it's quite controversial, interesting. I don't think this is the case. I perceive myself liquid biopsy as complementary approach to tissue biopsy. The problem lies in the fact that liquid biopsy is not universally applied to all cancer. It's not. It will be difficult to replace tissue biopsy, for example, because tumor biopsy provides you the material to perform histological assessment of the tumor. And this is the main approach for subtyping of the cancer. And again, in early cancer, the sensitivity of the detection using liquid biopsy is quite variable and low. So for those reasons, I think this should be treated as a complementary approach.
Thank you so much. We have time for a couple more questions. Our next audience member wants to know, what is the plasma input volume for Sera Extracta's cell-free DNA kit, and how does it correspond to the volume of blood needed?
Okay, so you can get around 4 mils of plasma from 10 mils of blood, and the kit has been validated for 0.5 mil input up to 4 mils.
Thank you so much. We have time for one more question. What is the average amount of cfDNA per 1 milliliter of plasma?
Oh, this varies greatly, and obviously it depends on the particular condition that a donor is in. But also it depends on the pre-analytical and analytical approaches. But if we concentrate on the condition of the donor, this will be in high picograms to maybe 15, 20 nanograms per mil of plasma in healthy donors, while in advanced cancer, this would probably go for over 100 nanograms per mil of plasma. However, I think there should be a bit of caution applied because, for example, after strenuous exercise, the amount of cell-free DNA in plasma can go also above 100 nanograms per mil of plasma.
Monika Seidel, thank you so much for your time today and for your outstanding presentation once again. Would you like to provide our audience with any closing remarks before we go?
I think I said everything that I want. I just want thank everyone for listening. And if you have any particular questions that I haven't answered or something was not clear during the presentation, please feel free to contact me and write an email to my email address that I believe was displayed at the end of the presentation.
Thank you so much. And I will ditto that statement. Thank you to our audience. And please know that any questions that weren't addressed at this Q&A will be addressed via email by the presenter. And this presentation will be available for on-demand viewing for 12 months. So please remember to share it with your colleagues who may be interested in today's topic. And don't miss out on other presentations on the agenda. Visit the agenda tab in the auditorium for a full listing. Thank you again for your participation. Until next time, stay safe and stay healthy. Take care, everyone. Bye-bye.
Thank you, everyone. Bye.